No.
So I want to start this, which is actually a little out of sequence for you guys. I want to start with a little bit of a broader explanation of reproductive function. And I don't remember if I showed you this picture in lecture or not, but this is a scanning electron micrograph of an egg cell covered in sperm, which I'm almost positive was generated through in vitro fertilization type techniques. So this is probably done in a petri dish. And it's a little misleading. There wouldn't actually be this many sperm cells on an egg at the point of fertilization, but it does show the relative size of the two types of cells. Now, talking about the reproductive system in lecture already, we've already talked about how eggs are formed, the follicle development and all that sort of thing. But what I want to talk about here is to sort of go back just a little bit further than that and sort of what the point of reproduction is and what the focus of what I'm covering for the reproductive system is on, which we kind of already did for the female side of the things in lecture. But when the egg cell here is fertilized, it's going to then divide into two cells like we see here. And then those two cells will each divide, making 4, and those will each divide, making 8, which will each divide, making 16. And this is just sort of following the doubling of the cells, the process of division. From 16 we go to 32, and then 32 to 64, 64 to 128. Not all of the cells are dividing exactly the same way, so they show these first 4 pictures of the doubling from divisions. And then these last 2 pictures are actually the same thing. One is suggesting sort of the outside appearance of this thing called the blastocyst, and then this is what it looks like sort of cut open. And the cells at this point are starting to differentiate into different groupings, and it's really pretty small, minor difference. This inner cell mass here is really what's going to become the embryo, and then the outer part, what's labeled the trophoblast, is going to become the structures in the placenta that interact with the mother's circulatory system. Tropho, that root is referring to feeding, basically. And so the trophoblast is the tissue that's a precursor to what's going to allow to feed the cell, feed the embryo. Now, really what I'm showing you this for is to lead up to the idea that all of these cells here and all of the cells in the process of dividing here are really identical at the genetic level. The DNA is the same. The fertilized egg cell duplicates its DNA and then divides, and then each of these cells we see here, the 70 to 100 cells that are suggesting in this picture, each nucleus here contains the exact same DNA. But we're starting to see some differentiation into different major cell groups. And that's going to continue on, eventually having all the different tissues that make up the body. Every cell in your body, with a few minor exceptions, have the exact same DNA, and what's making them different is simply what genes they use. So, for example, a cell in your epidermis is going to be using the keratin gene. A muscle cell will use the myosin gene, those sorts of things. And there are a lot more genes that are involved, but those are some proteins that you've learned about in this course, and so you can associate with those different things. But the cells have all the same DNA. How these cells divide is the process called mitosis, which is just your simple cell division kind of approach. And it results in two identical cells at the genetic level. So every nucleus looks exactly the same. Now, in any nucleus in the cells in your body, how many pieces of DNA are we going to see there?
Pairs.
How many pieces of DNA?
46.
46, right. And what do we call those pieces of DNA? Chromosomes, right. That number is actually indicative of the human genome. It's not the only thing about the human genome, but it does tell us something. So this picture, which is actually out of the last chapter in the book, which is about inheritance, but it's where that picture is. I just want to grab this to show the chromosomes. So here we have 46 chromosomes. This is what's called a karyotype, which is a picture made by looking at the chromosomes in a given cell. Right before mitosis starts, the DNA condenses into these discrete chromosomes that we can see. Normally, they're kind of all sort of jumbled together in the nucleus, but they're easy to see single things there. Also, what's not terribly obvious in this picture is that within each chromosome, there's actually 2 strands of DNA. So we just see this thick band here, but it really has 2 DNA strands that are linked in the middle somewhere, maybe right about there. And the 2 strands of DNA, I shouldn't say strands, the 2 copies of DNA, which are both double-stranded themselves, because DNA is a double-stranded molecule, they're just identical copies. And what's going to happen in mitosis is that one copy of the DNA is going to go into one cell, and the other copy is going to go to the other cell. Same thing with this. So the pairs of chromosomes that we see here are going to separate out so that the pairs are still going to be present in the resulting cell. You have a question?
Yeah. So the one line is 2 double-stranded DNAs. Helices or
right. So DNA as a molecule is a double-stranded, double helix, however you want to describe it.
Yeah.
Here we're just looking at this huge molecule that's all condensed together. We can't make out the strands. But within this, there's 2 pieces. And actually, for the sake of addressing that question a little bit better, let me go into the book here. So the chapter that we're dealing with, for the most part, about the systems, like what we did in lecture and what we're going to do after I finish talking about this stuff, is in this chapter on the reproductive system. But the following chapter here has some of this information. And am I in the right place? Maybe not. Let me actually jump back to the beginning of the book. I think the picture I want is actually in the cell chapter. Yeah, this is what I'm looking at. So this is a representation of what these chromosomes look like. The piece on this side is a chromosome, and then it's linked to a duplicate copy of itself. This structure is one of these things we see here. Okay, and so in mitosis, what's going to happen is one of these is going to go to one nucleus and the other is going to go to the other nucleus. So each of those cells have a copy of that chromosome. I really should put this picture in the presentation along with what I'm showing here. But yeah, that's what we have there. Now, we see here 22 pairs numbered, and the 23rd pair is designated XY. The 23rd pair is the sex chromosomes, which determine the gender. Having an X and a Y chromosome means that this particular cell comes from a male of the species. If it was 2 X chromosomes, which would look more like a matched pair, then that would be from a female. Women only have X chromosomes in that pair. Men have an X and a Y. And so women only contribute an X chromosome to their offspring. And gender ultimately is determined by whether the male contributes an X or a Y chromosome. Okay. But the point here I want to show is just that the chromosomes are numbered pairwise. So this is a pair of chromosomes number 1. One came from mom and the other came from dad. And then mom and dad's chromosomes 2, et cetera, et cetera. We can tell which ones go together. They don't look precisely identical because of the photo quality here, but we can see that there's some banding patterns, like these 2 dark spots here, these bands here, these bands here. So we know these 2 are both chromosome number 7 or 1 or 16 or whatever. So we can pair them up and see. Having 23 pairs of chromosomes is actually somewhat distinctive of humans. And to kind of illustrate that, I want to go to this picture, which is actually a cover of this American Biology Teacher magazine. But it's based on data from a study that's described in this magazine. And let me just zoom in here so you can see some more detail. What's going on here is 4 different species chromosomes are being compared. And these chromosome maps, we can see the banding pattern a little bit better. It gives us some information about where we are on the particular chromosome. So DNA in this area right here might be specific for a particular gene or something like that. This is just a map. But the banding patterns that are drawn in here are kind of similar to the banding patterns I just showed you in that karyotype. Those are actual chromosomes that have been stained. And if we compare 4 different species, we can actually see that their banding patterns are pretty similar. Like this area right here is almost identical across all 4 of these. The first one that we see in this group is human chromosome number 1. And then the second one, I'm pretty sure, is chimpanzee. The third one's probably gorilla. And then the 4th one is another great ape, bonobo or orangutan or something like that. And they're just showing that these closely related primates all have very similar looking chromosomes. There is some difference in some of them, like human chromosome number 1 doesn't match the other chromosomes number 1 because it has a little bit more stuck in here. So there's a bit more DNA. What actually happens with this DNA, I don't know, but it makes this end of the chromosome that much longer. Or whatever this is, if it's gorilla, they have an extra little bit of DNA here on the end. And an extra band there that kind of pushes everything down a little bit further. So they aren't identical, but they're very similar. In the group that's labeled chromosome number 2, and the number there comes from the fact that this is human chromosome number 2, in the other species, they don't actually have a chromosome that matches ours. This chromosome is very similar to the bottom half of our chromosome number 2, whereas this chromosome is very similar to the top half of chromosome number 2. And what that suggests is at some point, a common ancestor between humans and these other great apes, there is a divergence that 2 smaller chromosomes merge together, making the larger chromosome number 2, and the individuals that had that chromosome went on to become modern humans. Exactly when that happened, I don't know specifically, but that's what we can see here. So if we look at the picture overall, they're numbered 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 18. 17, 22, and this is the X chromosome, this is the Y chromosome, showing all of those different chromosomes for all these species. And there's quite a bit of similarity, but you can notice that there are a few little places that things are different here and there, which would correspond to differences that we see between the species. The reason I like to bring up this picture is it starts to introduce some ideas I want to talk about in how the reproductive system sort of ensures that we can reproduce new individuals of our own species. When I first saw this picture, it really reminded me of a show I watched, I think on the Discovery Channel 10 or more years ago, called Humanzy. And it was a 2-hour show that was mostly about this one animal that was found that looked like a chimpanzee, but also looked a little bit and behaved a bit like humans. And so they spent an hour and a half of this 2-hour show suggesting that maybe it's a human chimpanzee hybrid, hence the name Humanzy for the show. And then in the last half hour, they explained, haha, we've been straining along all the time, it's not a hybrid. And this picture actually tells us exactly why that's not possible. Humans have 23 pairs of chromosomes. Chimps and the other species represented here have 24 pairs of chromosomes. Our chromosome number 2 corresponds to 2 chromosomes in those other species. So we just don't have the same chromosome makeup as other species. When a sperm and an egg come together to bring one set of each of the 23 chromosomes to the fertilization, if the chromosomes don't match up correctly, it can lead to errors that will either not make fertilization possible, or the zygote won't survive, or implantation won't take place, or something like that. So for a big difference like this, it's just not possible for hybridization to take place. Chromosome number 2 is pretty sizable. Most likely, if a human chimpanzee hybridization were to take place, other than doing it, say, in a laboratory, if it were to have accidentally taken place, like this show is suggesting, it would be most likely that a human sperm were combining with a chimpanzee egg. And if that were true, then the human sperm would carry this huge chromosome number 2 that doesn't correspond to any of the chimpanzee chromosomes. And so the chimpanzee egg would sort of reject that, and it wouldn't fertilize properly. Now, that suggests why humans probably wouldn't hybridize with similar species. But it is possible for similar species to hybridize. And in fact, this picture here is an example of such a hybridization. This is an organism called a liger, which is the offspring of a male lion and a female tiger. Now, the male female aspect, what I just said, is important. If a male tiger and a female lion mate, then they produce what's called a tigon. And ligers and tigons are actually very different because of some genetic differences within the two species. I'm not positive, but I would assume that they are close enough species that they probably have very similar chromosomes. So they might have, and I'm making up this number, let's just say they have 33 pairs of chromosomes. So an egg and a sperm from a lion and an egg from a tiger, when they come together, the chromosomes might match up enough. But they're not going to make a completely viable offspring. Now, we can see very obviously that it's possible for them to make offspring, but I'm pretty sure that ligers are not able to reproduce themselves. And one of the reasons why hybridizations are kind of weird is actually representative in this. That's a really freaking big cat. Okay? And in fact, ligers are the biggest cat around. The reason for that is the gene that regulates body size, body growth, is found in the female lion genome, but in the male tiger genome. And so a liger gets its lion genes from the male and its tiger genes from the female, both of which are not carrying a copy of a gene that regulates growth. And so there's no gene to regulate growth, and these cats just keep growing larger and larger. There are other pictures which, for whatever reason, I chose this one to show, but there are other pictures that show ligers standing up on their hind legs, and they're like twice as tall as humans and that sort of thing. They're really big cats. Tigons, on the other hand, would be a lot smaller. That thing on the back is not a tigon. That's not how much smaller they'd be. They'd just be a small size lion or tiger, not like a house cat size. But they have sort of a double dose of that gene that limits growth, so they don't get that big. Because of the genetic differences between the species, they're not going to be entirely viable organisms to survive. Now, we don't naturally see ligers or tigons because this sort of thing has to happen in unusual circumstances, under unusual circumstances. This picture, I'm pretty sure, is taken from a big cat reserve in Florida or something like that. And the woman in the picture there's actually several pictures of ligers that she's often posing with them. Sometimes she's blonde, sometimes she's brunette. But so it's a thing that apparently where she works, they do, or maybe she owns the place, I don't know. But actually, lions and tigers don't exist in the same geographical space. So lions are mostly in the African continent, tigers are mostly in the Asian continent, and so they're not likely to meet up. It's only when humans have put them together, whether it's in zoos or parks or preserves like this or something like that, that's likely to happen. Now, there are other kinds of hybrids, and probably the most famous one is the mule. A mule is a hybrid between a horse and a donkey. The thing is that they are sterile. You can't make a new mule by taking two mules and putting them in a bedroom together and turning on Barry White. You have to get another horse and another donkey to make another mule. Whoever thought that up and whoever goes, hey, I need another mule, and decides to do that, I don't quite know, and I don't know why people want mules specifically rather than donkeys or horses. But they're a hybrid, probably because a donkey and a horse has fairly compatible genomes. They're just not compatible enough to make a viable offspring. There are also hybrids, for instance, between different canine species. One I didn't know of until I moved to this area is called a coy dog, which is a coyote and a dog hybrid. Now, that hybridization actually makes a viable offspring. So that suggests that coyotes and dogs are actually, in some sense, the same species. It's just they don't naturally reproduce because coyotes are in America and dogs are originally, I can't remember if it's African or European primarily, but humans brought them together. So dogs that we have as pets in this country, when they get loose, they can interact with the wildlife and hybridize there. But as far as I know, coy dogs actually do reproduce themselves. Well, sort of mix in with the wildlife that they've joined, and they become a regular part of that, suggesting that they're more biologically similar species. But geographically, again, they're not really. But I'm talking about hybrids mostly because people are kind of interested in this stuff sometimes. And also to illustrate that bringing an egg and a sperm cell together is not all that's necessary for actual reproduction. There's more in the process to make sure that viable offspring are produced. And there's no other species that humans are similar enough to potentially hybridize with. there's some recent evidence suggesting that modern humans and Neanderthals hybridized because there's Neanderthal genes that are being uncovered in the human genome, but no modern species is available for humans to hybridize with. So if anybody ever tries to tell you that there's such thing as a human chimpanzee hybrid, it's not true. And the one that was shown in this show I was talking about earlier, it's a weird looking chimpanzee. It doesn't look like other chimpanzees, and apparently it doesn't smell like other chimpanzees either. But and it also walks upright, so it looks very human in its behavior. But it just seems to be a weird kind of chimpanzee. There might be a little population of this sort of mutant chimpanzee somewhere, and this particular individual just got separated from the population. But other ones like it haven't been found, so not really sure exactly where it came from. But it could just be, you know, a weird variation on chimpanzee or something like that. Now, I started off here talking about how cells divide from the fertilized egg cell to make all the cells in your body, and that's through a process called mitosis, which is represented here. This is not a picture out of your book. For these presentations, I'm trying to put pictures from the book in, and if the book doesn't have it, I need to find a picture somewhere else. And this is, and also I'm trying to use pictures, if I'm taking from somewhere else, that are copyright free. And that's what this picture is, which is actually from Wikimedia.
Wikipedia?
No.
Wikipedia.
Wikimedia Commons, that's what I'm trying to say. Which is the repository of images that are used on Wikipedia. But those are all copyright-free images. And so this is one that represents mitosis. And I like this one, actually. There is technically a picture for mitosis in the book. There's not one for the next thing I'm going to show you. But the one in the book goes into some things that I didn't really need to put here. This is just showing how a cell will go through a process causing division, giving us two copies of the exact same cell that we had at the beginning. And in this, they're showing the cyclical nature of cell division here. And oftentimes what's happening in our body, one cell will divide and give rise to two cells. One of them will continue cycling through. The other one will end up specializing and not divide anymore. So a good example of this is in your epidermis. In the basal layer, the basal cells there are stem cells for the skin, and they'll divide over and over again. One of the cells out of each division becomes a keratinocyte that moves up through the layers of the epidermis. The other one stays in the basal layer and can keep dividing. So same kind of idea. They're representing the chromosomes here by these lines. They're long and short lines, and I would have personally chosen different colors, but there's a long and a short red and a long and a short blue. What happens right before mitosis is the chromosomes condense and duplicate the DNA into this sort of X-shaped structure, which I showed you earlier. And then it goes into division, and here just depicts sort of what's happening in division. The chromosomes line up, and then half of each chromosome goes to one side or the other of the spindle, making two nuclei that are the same as the original nucleus there. Now, the other type of cell division, which happens for the cells that fall into that group that I said, the minor exceptions to this, are actually the ones that we're interested in with this section. And they go through this process called meiosis. So these two pictures, one thing I like about them, they're obviously made by the same person. And they're pointing out that mitosis is cyclical, that cells keep dividing over and over again, especially stem cells. Whereas meiosis is not a cycle. We start with this cell and we end with four cells that are kind of like half versions of that cell. So we have the same kind of cell that has 4 chromosomes, 2 red, 2 blue, a long and a short of each. they duplicate their DNA, giving us these traditional X-shaped chromosomes. They line up a little bit differently. Instead of all four lining up right on the middle, they line up pairwise. So the two long ones and the two short ones get together. And I think for this picture, what they're suggesting is we have two pairs of chromosomes, the long pair and the short pair. The red chromosomes, let's say, are the maternal chromosomes, and the blue ones are the paternal chromosomes. And so they come together and pair up, and they can swap back and forth a little bit of DNA, which is an event called crossing over, which we see here. And then when the chromosomes separate, they separate pairwise, but not splitting the chromosomes just yet. So we have one long and one short chromosome in each of these cells as it divides, making half as many chromosomes, or segregated half as many chromosomes. But they still have the duplicated DNA. And we can see a little bit of red mixed in with the blue, or a little bit of blue mixed in with the red, because of this crossing over that's taking place here. Then each of these cells, which have half as many chromosomes as the original, divide again, and this is when the two halves separate out, and so we end up with 4 cells that have one long and one short chromosome in each. And then in this picture at the end, they have this cell pointing as it's joining up with something else that's not representing the picture, and together they make the zygote, which is what the fertilized egg cell becomes, the pictures I showed you earlier on. These are called gametes, the general name for the reproductive cells. If this is happening in the ovaries, then we're making egg cells, and if this is happening in the testes, we're making sperm. I talked a little bit more about specifically what's happening in the ovaries in lecture. So we don't make 4 equal cells. I don't make any egg cells. But in the ovary, it's not making 4 equal egg cells. It's making 1 egg cell, and the other 3 are the polar bodies that are essentially just throwaway cells. Producing sperm, the 4 are equivalent, and they're going to each make sperm in the process. So really the point of talking about the reproductive system, what I did in the lecture before and what I want to do today, is to really think about where this process takes place and how those cells come together. So we've already done that with the ovaries and the female reproductive system. Today I just want to concentrate on the male reproductive system. So we're going to look at the structures that make up the male reproductive tract in these pictures here. So starting with this picture, and these are mostly pictures out of the book, I just want to talk through the structures that are part of the male reproductive tract. And I want to do it in the order from where the sperm are produced to where they travel and eventually leave the male reproductive tract. And then I'll go back and look at some of these things in a little bit more detail. So it starts with the testis, which we see here. We'll look at more detail of what's going on in the testis, but this is the gland where the sperm are actually made. The sperm leave the testes and move into what's called the epididymis, which we have here, sort of wrapped around from the top of the testis to the bottom. And then the sperm leave the epididymis and go on into this tube here, which leaves the scrotum and enters into the pelvic cavity. And that tube is the ductus deferens. Now, you've probably heard this referred to more often as the vas deferens, but that's actually an incorrect name for it. Vas means vessel, like a blood vessel or a lymphatic vessel, which implies something that's not true for this. So ductus deferens is more appropriate because it's really a duct. What's incorrect about calling this a vessel is what epithelium makes up the wall of a vessel. What epithelium do we find in the wall of a blood vessel or a lymphatic vessel? Simple what?
Squamous.
Right, simple squamous. Thinnest epithelium you could have, which is great for things that are diffusing across the epithelium, like nutrients or waste products or gases or something like that. But for a duct, we don't want that. So a duct has a different epithelium. And depending on this specific duct, it can have different types of epithelium. The male reproductive tract is lined with a pseudostratified epithelium. Other ducts might have a simple cuboidal or simple columnar, or even a stratified cuboidal or stratified columnar, but here it's pseudostratified. Actually, what we're looking at here, what's labeled as a ductus deferens, is more than just the ductus deferens. It's surrounded by some supporting tissue, connective tissue, and that sort of thing that holds it in place. And collectively, all of that's called the spermatic cord. And that goes over the over the pelvic bone and into the pelvic cavity, and then runs alongside the bladder, kind of hooks over the ureter, and then comes around posterior to the bladder, where it meets up with the next structure called the seminal vesicle. And where the seminal vesicle and the ductus deferens come together, they form the ejaculatory duct, which passes through part of the prostate before it joins up with the urethra. The urethra is part of the urinary tract. It's leaving the bladder, passing through the prostate also. When the ejaculatory duct and the urethra meet up, from that point on, the urethra is part of the reproductive tract as well as the urinary tract. It leaves the prostate and then enters into the penis. This represents the base of the penis. The urethra continues on along the shaft of the penis to the end where it opens at the external urethral opening. Where we see the urethra coming in, it's joined by another duct coming from what's called the bulbourethral gland. And the urethra is passing through the penis, surrounded by this tissue, kind of represented by this mottled red colored area here, called the corpus spongiosum, which is very similar to more tissue that we see. It's a weird representation of this tissue. You really don't see too much of this actually right on midline, but they're trying to depict that it's running along the length of the penis also, called the corpus cavernosum. There's actually two of these things that are called the corpus cavernosum here, and the plural of corpus cavernosum in Latin is corpora cavernosa, which we'll see in another picture later on. But both of these things that are called corpus something or other are erectile tissue, and they're depicted in this mottled red because they have a lot of blood vessels, and as the blood fills that tissue, that's what causes the erection. When the blood gets into this tissue and starts to cause erection, it's going to push on the bulbourethral gland, which will secrete fluid that gets into the urethra and helps to sort of clean it out, neutralize and clear out any components of the urine that might be there still, in preparation for when the sperm come along. And the sperm, when they get to at least the prostate gland, are going to mix with fluids coming from the seminal vesicle as well as fluids coming from the prostate, which collectively is called the ejaculate. It contains fluids which will further play a role in neutralizing acids found along the length of the reproductive tract. Actually, not just the male reproductive tract, but also the female reproductive tract will have some acidic secretions. In both situations, it's for inhibiting microbial growth. So the sperm needs to have the ability to, or not the sperm, but the ejaculate needs to have the ability to neutralize those acids. Also, the sperm need energy to swim, so there's some simple sugars present in there, fructose mostly, which
provide the energy-rich molecules that mitochondria in the sperm can use to swim. So those are the structures, and I want to go through and look at a few things in more depth, like the testes, the seminal vesicles, and the erectile tissue of the penis, with some other pictures. Before we get there, though, this picture is just showing the testes and epididymis and the beginning of the ductus deferens are found within the scrotum, which is an extra pelvic position. Also, besides those structures I talked about for the tract, there are some muscles, the cremaster muscle and the dartos muscle, represented here, which are capable of moving the testes up or down closer to or farther away from the body wall within the scrotum. A lot of sources, and probably the book, will say that the purpose for that is temperature control. Spermatogenesis, the process of making sperm cells, the meiotic divisions and everything, needs a temperature of about 34 degrees Celsius, and body temperature is about 37 degrees Celsius. So they need to be a little cooler. And so the suggestion is, especially because of these muscles, the testes can descend away from the body wall to stay cool, or move towards the body wall to be warmed up, depending on the ambient temperature around. The problem with that actually is that it doesn't really explain species that have internal testes, which have the same temperature issue to play with. And I read a study a couple years ago now that suggested really probably the point of the testes being extra pelvic, which is really pretty dangerous, because if something happens to the testes, it makes it impossible for reproduction to take place. And that kind of pressure on reproductive structures should always sort of err in the side of keeping things protected so that the species can reproduce. But probably the reason that they're extra pelvic has to do with the blood vessels that we see here. For species that run, and I'm paying most of the attention to mammals. So as they run, the hip joints are going to be moving quite a bit. And if all of this is internalized, these blood vessels are going to be right across the hip joint. And they can be stressed, torn, damaged, whatever, in that process. So by moving these outside, it protects the blood supply, which is probably more important than making the testes completely secure inside the body. Okay. So if we look inside the testis, we see it's actually a bunch of tubes. And these are called seminiferous tubules, which are contained within the testes, sort of surrounded by a layer of tissue, this tunica albuginea. I can never pronounce that word. And it makes the testis seem like a solid gland, but actually it's a bunch of tubes. The first person to successfully publicly dissect this, and back in that time, medical schools had sort of events where professional anatomists would come in and dissect things for everybody to see. So there's a guy that very carefully cut through the outer covering here, and all of these tubes just spilled out, and everybody was very surprised with that. Because up to that point, most people just assumed the way reproduction happened is that inside the testes were these little itty-bitty versions of human beings, which were then passed to the female, where they would then grow as the embryo and fetus, that sort of thing. And all of the contributions to producing the new human being really came from the father. That was one way of sort of picturing how this all worked, which was obviously very wrong. But that's the way people thought, because obviously men were in power, so they had to be the important part of reproduction. And so nobody expected to see tubes like this spilling out. Those tubes are clustered together between these septa, and eventually make their way to this area here called the rete testis, where the tubes kind of condense together or come together. And then they feed into the epididymis, which we see here. And the sperm will then travel through the epididymis down to its tail, turning around this corner and eventually getting into the ductus deferens, which will carry them out of the scrotum and into the pelvis. If we look at a cross-section of one of these tubes, we see that pseudostratified epithelium here, which is not so obviously a pseudostratified epithelium, but part of the point of calling it pseudostratified is that it's not that easy to tell. But actually why it looks a little unusual is because there's a bunch of layers of cells, not epithelial cells, but rather the cells that are going through meiosis to produce sperm that are worked into the epithelium. The actual cells that make up the epithelium, which are called Sertoli cells, there's only one Sertoli cell that's the width of this epithelium, so it's pseudostratified. So this is a picture representing how sperm cells are made. There's a similar picture which I showed you in lecture for how egg cells are made. One little difference here is instead of just saying N representing the 23 chromosomes that are present there versus 2N for the 46, they say 1N. Mathematically, N by itself and 1 times N are the same thing. But it's just a little quirk. You don't usually see that. And in fact, in the comparable picture for the production of the egg cell, the book uses just N. So I really don't know why they put 1 there, but they did. And this shows the mitotic divisions of the stem cells that make the primary spermatocytes, which then divide into the secondary spermatocytes and then divide again, giving rise to the 4 spermatids, which then undergo a process to become the specialized sperm cells. Now, going from spermatid to sperm cell itself is called spermiogenesis. The whole process, including the meiotic divisions, is called spermatogenesis. Okay. So those two terms can sometimes get confusing, but spermatogenesis is the whole picture here. Now, the sperm cell is specialized. The gamete that's produced at the end, that spermatid, all that's important about that is its nucleus and the DNA that's in there. It loses a lot of its cytoplasm, and all that's left over are specialized structures like this acrosome that we see surrounding the whole thing, and then a bunch of mitochondria which are wrapped around the base of a flagellum and centrioles that are necessary for nuclear processes that are going to happen after fertilization. All of these mitochondria are there to make the ATP necessary for the flagellum to spin around and therefore the sperm cell to swim. I mentioned a little bit ago that secretions from the seminal vesicles and or prostate provide simple sugars to power ATP production. So these mitochondria use, I think it's actually fructose, not glucose, to do that, but it's the same basic process. Okay. And it's really just a DNA delivery device. Nothing about the sperm cell persists other than the DNA after fertilization. All of the mitochondria, did I talk about this in lecture? All the mitochondria in egg cell came from the mother? No, I didn't. Okay. I probably didn't because I hadn't talked about sperm yet, but the mitochondria in the sperm don't go into the egg cell. All of the mitochondria that are in the sperm are from the mother, and all of the mitochondria in the mother came from the grandmother, and all of the mitochondria in the grandmother came from the great-grandmother. Mitochondria have only a matriarchal descent. And the mitochondria from the sperm just don't get introduced there. And that kind of corresponds to the idea that I did talk about in class, that the egg cell ends up being this huge cytoplasm-rich cell, and the mitochondria are in that cytoplasm. Okay, nothing's contributed besides the DNA from the sperm. This picture is not in your book. It's actually from Gray's Anatomy, not the show, but the actual anatomy book. And Dr. Gray, again, not Meredith, but the real Dr. Gray, drew pictures of a bunch of different things. The book is so old that all of the pictures are in the public domain. They're not copyrighted anymore, if they were ever actually copyrighted. But they're very good representations. And I pulled this picture out because there's not something like this in the book showing the seminal vesicle and the ductus deferens where they come together. They actually kind of come together right at the last minute where they give rise to the ejaculatory duct that passes through the prostate to meet up with the urethra. And the same thing happens on the other side. The end of the ductus deferens, which in Gray's Anatomy is still referred to as the vas deferens, is called the ampulla. It kind of thickens out a little bit, has this sort of big chamber here. And it meets up with the seminal vesicle, which we can see here, which is making some of the seminal fluid secretions. And that ends up getting into the into the urethra to be carried on along. The prostate also secretes things that get into that seminal fluid also. And we can see the prostatic urethra there. The erectile tissue that I mentioned before, which we can see in the mid-sagittal section like this, actually looks more like this in terms of a cross-section of the penis. Surrounding the urethra is the corpus spongiosum, which looks like it's most of the tissue in this picture, but it's actually a fairly small contributing part of that. And then more of the erectile tissue are these two big oval structures here, which are plural in the plural referred to as the corpora cavernosa. A single one is called a corpus cavernosum, which is what the earlier picture showed. And on midline, they just show a little bit here because there really isn't that much right on midline. Instead, it's mostly off to either side, but it makes up most of the thickness of the shaft of the penis. There's an artery running right through the middle of either one, bringing blood into this tissue. And when that happens, all of the blood vessels in this tissue have dilated, allowing the volume of blood to be quite great. And that's what leads to the erection, engorging of this tissue, which I think is sometimes referred to as tumescence, another word for the stiffening of the tissue. And it comes from that particular erectile tissue there. Before I go there, so that allows for the urethra to deliver the sperm to the female reproductive tract. And just, I don't have a picture to depict this, but I have this model of the female reproductive tract, and we can see the sperm that are coming in here. So they've been delivered from the male reproductive tract. They move through the vagina and the uterus, and they just randomly go left or right here. They don't necessarily know where to go. Some of them will end up making their way into the uterine tube, where they'll meet up with the egg cell that's been released at ovulation. These are not drawn to scale. An egg cell and a sperm are not as big as suggested relative to the organ size here, but this is where fertilization takes place. And then after fertilization, that fertilized egg cell divides into two. There's 2 divided into 4, 4 into 8, 8 into 16, et cetera, et cetera. Eventually, the blastocyst that I showed you earlier implants into the endometrium of the uterus. And the cell mass that's on one side of the blastocyst is what's going to become the embryo. And the trophoblast that I pointed out earlier is what's going to become the placenta that interacts with the uterine wall to provide the nutrition to the developing embryo.
So I just wanted to show you that with the model, just to show you how the male reproductive tract fits with the female reproductive tract. This picture out of the book is showing the connection of the hormones. Now, did I show you this at the end of class on Thursday? Maybe not. I might not have in class because I was saving it for lab where we're talking about this stuff. But in class, we did talk about growth gonadotropin releasing hormone, which is correctly abbreviated in the caption up here, but for some reason the H and the R got changed. It's GnRH, more appropriately, for gonadotropin releasing hormone. That comes from the hypothalamus, which tells the anterior pituitary to release FSH and LH. Exact same thing for the female system that we talked about in lecture. FSH for the male system stimulates Sertoli cells, which promotes spermatogenesis. Those are the nurse cells, or the sustentacular cells, I think is what the book calls them. But they're supporting cells there in that seminiferous tubule wall, and they promote the division of the cells through meiosis to make sperm. LH targets cells in the connective tissue between the seminiferous tubules called Leydig cells, which then produce testosterone. And it's not represented in this picture, but testosterone will also stimulate Sertoli cells to promote spermatogenesis. But what they're showing here is that testosterone feeds back to the anterior pituitary and the hypothalamus, just like estradiol does in the female system, to limit the release of those gonadotropins. They've added in here, and why they didn't show it in the female system, which we were talking about in lecture, they've added in the release of inhibin, in this case from the Sertoli cells, which also inhibits pituitary hormone release. In the female system, inhibin is present also. It's being released from the follicles as well. But for whatever reason, the book, they left that out because it's really kind of redundant with the estradiol or testosterone signal we see here. But it adds to that. So in the male system, the exact same hormones are used. We don't need to bother about FSH and LH's names because those names don't apply here. Follicle stimulating hormone applies to follicles being stimulated to develop in the ovaries. Luteinizing hormone refers to the corpus luteum that forms in the ovaries. Neither of those things are happening here because we're not looking at ovaries, but it's still the same hormone. It's just used slightly differently. Still to promote the production of the reproductive gametes, but it's just a slightly different system here. There isn't the same kind of luteinizing hormone dependent spike that causes ovulation in the ovaries. There's nothing like ovulation here. This process is just always going on. There might be some cyclical nature to this. I've not researched in a lot of detail, but there is every so often reference to the male reproductive cycles. So there might be times, and it might be a 28-day cycle or something like that. I have no idea. There might be times where sperm production is higher because of hormone levels, but it's not really quite the same as what's going on in the female system. So it's not really tied so much there to a longer cycle. But we see the same players here having an effect on the male system. So that's actually all I wanted to cover for the male reproductive system. In the practical that you'll be having next week, the case is about prostate gland enlargement. And because the male system combines urinary and reproductive tracts through the urethra, That case is really getting at both the urinary and the reproductive system material. And in this model, which I showed you with the urinary system last time, this purple thing here at the bottom is actually a prostate gland. So we're looking at the male reproductive system. Here are the ureters, because this is urinary model. These are the spermatic cords with the ductus deferens in the middle, which are kind of hooking over the ureter when they meet up with the seminal vesicles, which we see here. So this model is actually of the male system, because we see the prostate and the ductus deferens and the seminal vesicles there, that are the reproductive structures that are associated with the urethra for the male reproductive tract. So that case kind of puts the urinary and reproductive systems together to address both of them. There's 4 cases. Actually, I don't quite know why I did this exactly, but there's the respiratory system case, there's 2 digestive system cases, and then there's 1 case that covers urinary and reproductive systems together. I got 4 cases, I got 4 systems. I don't know why I didn't make them each about each other. The 2 digestive system cases, basically one's focusing more on the upper digestive tract and the other's on the lower digestive tract. So covering that much of the digestive system, I wanted to use 2 cases there. But so the other 2 systems are kind of both addressed in that case there. So again, focusing on the male side of the system in lab corresponds with the material for the practical that you'll have there. So, are there any questions about male reproductive system material?
I have a question.
Yeah.
Is this the room that we go to for the?
Yeah, we'll be in here. And you guys,
12.
yeah, we'll come in at 12. Technically, the exam period is 11 to 1:50. No, 11 to 12:50. But you'll have, since you're starting at 12, you can stay till 1:50 if you need, so that you have the full hour and 50 minutes. I doubt any of you have another exam right after that, but if you do, let me know and we can deal with that. But it should work out that there will be time for everybody.